Okay, so welcome to this next video in which we are discussing diabetic retinopathy. So what we've discussed is that in diabetes, what you have is chronic and persistent hyperglycemia. So you have times where your blood sugar is far too high and they are very, very regular and they last for extended periods of time. Okay, and this high blood glucose is not good for the blood vessels in the retina, basically, and it causes diabetic retinopathy. Now, the first stage of diabetic retinopathy is pre-proliferative diabetic retinopathy. And we've talked about three of the things which are characteristic changes that occur due to the hyperglycemia. Okay, so we've discussed that the number of pericytes that you have surrounding your capillaries vastly reduces. The thickness of the basement membranes of the microvasculature hugely increases, and you also get microaneurysms, these dilatations of the blood vessels that are pathological. Okay, now those aren't the only three things, so let's continue on. Now, importantly, what happens is the capillaries become much more leaky. Okay, so basically the endothelial cells of capillaries, they don't they don't make up the entire capillary. You'll have other endothelial cells and there will be junctions between neighboring endothelial cells. So let me draw another picture for this. Okay, so permeability is going to go up. Okay, so let me draw another picture. So I'll move that piece of paper out of the way. Right, so let's draw a capillary from the side view, okay, rather than in cross-section as we have done before. So let's say this is our capillary here. Okay, so it's a tube, okay, fundamentally, and we'll draw the endothelial cells. So basically, you might have one endothelial cell here, and let's say this is its nucleus. You might have another endothelial cell here, another one here, like so, another one here, okay, and it continues on like this. Now, basically, there are junctions between neighboring endothelial cells. So, for instance, if I just highlight this in, here is a point where two endothelial cells meet. Okay, now, usually, in the retina, there is something known as a blood-retina barrier, which is a very similar concept to the blood-brain barrier. So basically, it's not true of all capillaries in the body, but these junctions between endothelial cells in capillaries within the retina are going to be very, very tight. Okay, so in other capillaries elsewhere in the body, there might be nice big gaps between neighboring endothelial cells and the capillaries will be very leaky. But in the retina, there is this concept of the blood-retina barrier. Okay, and basically this means that these junctions between the endothelial cells are going to be extremely tight. So you can't just move across that membrane, basically. Fluid is not just going to seep out from the capillary lumen into the interstitial fluid. Nutrients can cross either because it will diffuse across in the case of oxygen or carbon dioxide, or because there will be transport mechanisms in the case of glucose that transport it across the endothelial cells. Okay, but fluid can't just move from the blood and seep out into the retina. Okay, now in diabetic retinopathy, what happens is the blood-retina barrier kind of breaks, basically, so it becomes lousy, and the gaps between the endothelial cells open up. Okay, so let me draw some of this. So here we've got these beautiful tight adhesions between neighboring endothelial cells. Maybe after diabetic retinopathy, what's going to happen is your capillary will start looking something more like this. Okay, so you'll have an endothelial cell here. The next one might be over here. So you've now got these leaky gaps that we can see here between the endothelial cells. Okay, so basically the blood-retina barrier is going to break down and these capillaries are going to start looking like the capillaries that are in other tissues of the body rather than the capillaries that should be in the eye. Now, what's the significance of this? Well, fluid is now going to be able to come from the blood and seep out into the interstitial space, okay, and it's going to form what's known as an exudate. and this is going to cause the swelling of the retina, basically. Now, the specific portion of the retina that is most affected is the outer plexiform layer. So let me get my picture of the retina back again. So basically, the portion that you notice the most swelling in is the outer plexiform layer, basically. Okay, so you're going to get fluid accumulating in here, and it will get thicker, basically. Okay, in addition, basically what this increase in permeability is going to cause is it's going to cause something known as macular edema. Okay, now edema just means the accumulation of fluid, basically. So of course we are going to get edema because we have this exudate forming, and basically macular edema just means the accumulation of fluid in the macula. And basically when you do an ophthalmoscopy, okay, so you use an ophthalmoscope and look into the back of the eye of someone who is suffering from diabetic retinopathy in the pre-proliferative stage, what you will see is swelling of the macula. Basically, you can see the accumulated fluid there. Okay, and that's called macular edema. So this increase in permeability is going to lead to an exudate forming in the outer plexiform layer and also to macular edema. Okay, right. Let's now talk about one final thing. So this was the fourth thing, that permeability is going to go up, and that's going to lead to macular edema and an exudate forming in the outer plexiform layer of the retina. Okay, now the fifth thing is that you also get little occlusions forming in the microvasculature. So you get little blockages where blood can no longer flow through, and the cause of these isn't really well understood, but it is noted that there are occlusions basically in the blood vessels. Okay, so all of these things now are going to lead to the blood supply to the cells of the retina going down. So the occlusions, it's quite easy to understand how that's going to lead to blood supply going down. If you've got a blood vessel that is occluded, then there's no blood going down further past the occlusion, okay? And therefore all the tissue that this further down blood vessel supplies aren't going to be getting nutrients. But also the formation of the exudate isn't going to help either, because what that will effectively do is it will become a sort of stagnant, stable pool of fluid, and basically it will push a gap between the blood vessels and the tissue, basically. So if you imagine, if I get my picture of the outer plexiform layer back again, okay, if you imagine a huge amount of fluid coming in here, what's that going to do? Well, it's going to increase the diffusion distance between the blood vessels and the tissue, basically, okay, and it's going to lead to actually impaired exchange between the blood and the tissue. So basically what all of these things in the pre-proliferative phase of diabetic retinopathy lead to is it leads to the tissues of the retina becoming hypoxic. Okay, and this is now why we're going to lead to, well, this is what's going to cause the proliferative stage of diabetic retinopathy. So you start with hypoxia of the tissues of the retina, and this is due to all of the things that we've discussed in the pre-proliferative stage. So, for instance, the microvasculature aneurysms that we've seen, those won't help blood flow through the area. They'll lead to improper flow, which will limit blood supply to that area. Okay, so all of these things will contribute to the retina tissues becoming hypoxic. Now, when retina tissues become hypoxic, what do? Well, they call for help. Okay, they call for more blood vessels to be built. They call for the beginning of angiogenesis. So these hypoxic tissues will start releasing vascular endothelial growth factor A. There are other forms of vascular endothelial growth factor, but VEGF-A is the main form that is responsible for hypoxia-induced angiogenesis. Okay, so this stands for, the V is for vascular, the E is for endothelial, and then the GF is for growth factor. So this is vascular endothelial growth factor A. Now, there are different isoforms of vascular endothelial growth factor A, but we won't worry about those here. Okay, so what will happen is the retinal tissues will start releasing vascular endothelial growth factor A, and what this will trigger now is angiogenesis. So it will trigger both sprouting and intussusceptive angiogenesis. Okay, so let's talk about the two different forms of oops, it's not going to fit in there, never mind, angiogenesis. Okay, so there's two major forms of angiogenesis that are known. The main one, the one that everyone thinks of when they think about angiogenesis, is actually called sprouting angiogenesis. Okay, so this is basically where you have an existing blood vessel here, let's say, and then you have some sort of angiogenic center here which is releasing VEGF, okay? So this is an angiogenic center, somewhere that is hypoxic basically, okay? And it's releasing VEGF-A, so here comes the VEGF-A, okay? And this will act on the blood vessel, and it's usually a capillary that will actually do sprouting angiogenesis. And what it will trigger is it will trigger the capillary to basically produce a sprout which will migrate towards the source of the vascular endothelial growth factor A. Okay, so you'll produce this new sprout basically off the side of your blood vessel, and basically if you've got another blood vessel, another capillary doing the same thing over here, what you'll get is these multiple sprouts all converging on the angiogenic center here, and basically when they meet, these two sprouts can then fuse to establish a link between these two separate capillaries here. Okay, so that's sprouting angiogenesis. Intussusceptive angiogenesis is quite different from this. So intussusceptive, a big word. Okay, intussusceptive angiogenesis basically is also called splitting angiogenesis, and that basically describes what it's all about. You take one blood vessel like this, and when it undergoes intussusceptive angiogenesis, what will happen is basically it will split into two separate blood vessels like this in the middle. Okay, so it goes from being one blood vessel to being these two separate tubes here. Okay, and that can actually occur in bigger blood vessels than just capillaries. Okay, so these are the two forms of angiogenesis that vascular endothelial growth factor A can trigger. Intussusceptive angiogenesis, there's hardly anything known about that. Pretty much we're still debating whether it even exists. We have pretty much settled that debate now. We do think it exists. But sprouting angiogenesis, we've known about this for over 200 years, so much more is known about that. And generally this is the form that people think of when they think of angiogenesis. Okay, so let's see what's going to happen then. And we are now going to go into what's called the proliferative phase of diabetic retinopathy. So this is proliferative diabetic retinopathy. Okay, and the reason it's called the proliferative diabetic retinopathy is that to produce these new blood vessels, of course, cells are having to proliferate. So this is the proliferative phase, basically. Okay, so this is really going to all result from where you're producing these new blood vessels. So basically we need to discuss where these new blood vessels are actually going to be produced. So basically what happens is most of these blood vessels end up being produced in this sort of nerve fiber layer. Okay, and what happens is firstly the sprouts start to breach into the optic disc. So what basically, if you look at someone with in the proliferative stages of diabetic retinopathy, if you look at the back of their eye using an ophthalmoscope, what you'll see is little sprouts of blood vessels growing in on the optic disc. Okay, so you'll see new blood vessels breaching into the optic disc. Okay, so that's the first thing that you'll see. Now, when you have blood vessels breaching into the optic disc, that is known as disc neovascularization. So when you get new blood vessels being formed, it's called neovascularization. And because they're breaching into the optic disc, it is called disc neovascularization. Now, more important than disc neovascularization, what is also going to happen is some of these blood vessels will penetrate through this internal limiting membrane. So basically, let me draw a separate picture for this. Okay, what you'll get is blood vessels now pressing this internal lining membrane. Okay, so here it is. So this is the internal limiting membrane here. Okay, and you'll now have blood vessels that are coming out and breaching and going in on the inner layer of this. Okay, so here's a new blood vessel. And you won't just be forming one of these, you'll be forming many of them. So basically it forms a whole meshwork on the surface of the internal limiting membrane. Okay, so basically when you have this new blood vessel formation upon the internal limiting membrane, what that is called is it's called retinal neovascularization. So basically you're getting these new blood vessels formed on the surface of the internal limiting membrane, and this is called retinal neovascularization. And again, if you look at the back of someone's retina with proliferative diabetic retinopathy, you'll be able to see this new meshwork of blood vessels on the surface of the internal limiting membrane. So basically what you'll see is just meshworks of blood vessels all over the place, basically like so. Okay, so let me try and show this. So here is a meshwork. Now let's think about where this meshwork actually is. Well, it's on the internal limiting membrane, but what faces the internal limiting membrane? Well, let me get my picture of the retina back again. So remember, the internal limiting membrane is the internal aspect of the retina, and it faces onto the vitreous chamber. So basically, usually the vitreous humor, which remember is this huge piece of jelly that sits in the vitreous chamber, is pressed against the internal limiting membrane. So basically you're forming all of these new blood vessels, this mesh of blood vessels between the limiting membrane, the internal limiting membrane, and the vitreous humor, basically, in that potential plane between the two. Okay, and this meshwork of blood vessels that you form is what's known as a neovascular membrane. So this web of blood vessels is called a neovascular membrane. Now, why does this cause problems? It causes a huge number of problems, basically. Now, one of the key horrible things that can result from this is basically these new blood vessels which you form, they're not particularly brilliantly intact, basically. They are very, very leaky. Now, usually it's not a problem because when they're initially formed, remember, they are pressed between the internal limiting membrane and the vitreous humor. So basically, because they're pressed in between there, the fact that they are leaky doesn't matter because they're surrounded by the vitreous humor on one side and the internal limiting membrane on the other. That kind of keeps the blood inside, basically. But if the vitreous humor moves away from the internal limiting membrane, okay, so-called posterior vitreous detachment, then what can happen is you can get huge hemorrhage, basically. So if you get posterior vitreous detachment, okay, which is when the vitreous humor moves away from the internal limiting membrane, okay, then what can happen is that these newly formed blood vessels, this neovascular membrane here, can end up hemorrhaging, okay, so you can leak huge amounts of blood, and obviously that will lead to severe visual impairment. Right, so that's one of the horrible, nasty things that can occur because of this neovascular membrane. In addition, however, the neovascular membrane, the formation of it can also actually lead to the wrinkling of the retina, okay, which again can lead to visual impairment. So if the retina is no longer a smooth surface, instead if it's wrinkled, that will obviously distort the angle that the photoreceptors are at, and therefore their capacity to receive light from certain directions will be affected, basically. Okay, so that can also lead to visual distortion. But most scary of all, what it can lead to is something known as retinal detachment. So the formation of this neovascular membrane can lead to what's called retinal detachment. And basically, retinal detachment is when the photoreceptors move away from the retinal pigment epithelium. Okay, so remember our original picture of the retina here. We had the retinal photoreceptors sitting on the retinal pigment epithelium. But what this neovascular membrane can lead to is it can lead to the detachment, the cutting off here effectively, and all of this lifts away from the retinal pigment epithelium. Okay, and obviously when the photoreceptors are no longer in contact with the retinal pigment epithelium, they're not going to be able to function anymore, because remember what the retinal pigment epithelium did. It regenerated the pigment every time. What was happening is the visible pigment was moving from these rods and cones to the retinal pigment epithelium to be regenerated, and then it was being sent back to the rods and cones. So if you no longer have the retinal pigment epithelium in contact with the rods and cones, they can't function in their phototransduction activity anymore. So it can lead to blindness, basically. Okay, so this is how basically diabetic retinopathy can lead to blindness, and it is one of the leading causes of blindness, certainly in the Western world, okay, because of the obesity epidemic, as it's being called. Okay, so that now concludes our discussion of the pathology of diabetic retinopathy.